Chapter 3 Plant Kingdom. In the previous chapter, we looked at broad classification of living organisms under the system proposed by Whittaker in 1969, wherein they suggested the five kingdom classification Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within Kingdom Plantae, popularly known as Plant Kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the members of Monera and Protista having cell walls have now been excluded from Plantae. Earlier classifications placed them in the same kingdom. So the cyanobacteria that also referred as blue-green algae are not algae anymore. In this chapter we will discuss about algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influenced the classification systems. The earlier systems of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based on mainly on vegetative characters or on the andrecium structure system which is given by Linnaeus. Such systems were artificial. They separate the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment as against these natural classification systems developed which were based on natural affinities among the organisms and considered as not only the external features but also internal features like ultra structures, anatomy, embryology, phytochemistry, such as classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relations between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These are become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Numbers and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundred of characters can be considered. Cytotaxonomy, that is based on the cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plants to resolve confusions are also used by taxonomists these days. Algae. Algae are chlorophyll bearing simple thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and wood. Some of them also occur in association with fungi known as lichen and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonical form like volvox and filamentous form like olothrix and spirogyra. A few marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. The vegetative reproduction is by fragmentation. Each fragmentation develops into a thallus. A sexual reproduction is by production of different types of spores. The most common being the zoospores. They are flagellated, motile and on generation gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated, similar in size as Ulothrix, and non-flagellated, non-motile but similar in size as in Spirogyra. And next, such reproductions is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudonaria is termed as anisogamous. Fusion between one large and non-motile static female gamete and smaller motile male gamete is termed as oogamous. Example Olvax and Fucus. The pictures here we are seeing are belongs to the green algae. The colonical one is known as Volvax and the second one is Ulothrix. Now about brown algae, you can see the first one named as Laminaria. You can see the labeling here, and as well as second one is Fucus. You can see the midrib and hohlfast, and the third one is Dictyota. Now coming to examples of red algae, here you can see that Porphyra as the first one and the second one is Polysiphonia. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra and Laminaria and Sargassum are among 70 species of marine algae are used as food. Certain marine brown and red algae produce large amounts of hydrochlorides, water holding substances, example algin, brown algae, and charazin, red algae, which are used commercially. Agar, one of the commercial products is obtained from Gelidium and Gracilaria, are used to grow microbes in preparation of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae is divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonical, or filamentous. They are usually grass green due to dominance of pigments chlorophyll A and B. The pigments are localized in defined chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have rigid cell wall made up of an inner layer of cellulose and outer layer pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different type of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangiae. The sexual reproduction shows considerable variation in type and formation of sex cells. It may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvax, Ulothrix, Spirogyra, and Chara. Phaeophyceae. The members of Phaeophyceae are brown algae are found primarily in marine habitats. They show great variety in size and form. They range from simple branched filamentous form ectocarpus to profusely branched form as represented by kelps which may reach heights of 100 meters. They possess chlorophyll A and C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophylл pigment. Fucoxanthin present in them. Next. Food is stored as complex carbohydrates, which may be in the form of laminaria or mannitol. The vegetative cells may have cellulosic wall, usually covered on outside by gelatinous coating of algin. The protoplast containing in addition to plastids a centrally located vacuole nucleus. The plant body is usually attached to substratum by holdfast and a stack and a stipe and a leaf-like photosynthetic organ the front. Vegetative reproduction takes place by fragmentation. Asexual reproduction is most brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water within the oogonomous species. The gametes are piriform, pear-shaped, and bears two laterally attached flagella. The common form are ectocarpus, dictyota, laminaria, sargassum, fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrid in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in the oceans which relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in the structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Now let's see the table division of algae and their main characteristics. Classes. First one, chlorophyceae. Common name is green algae and majority pigments are chlorophyll A and B. Store food is starch. Cell wall is made up of cellulose and the flagellary numbers are 2 to 8 and the positions are equal and apical. Habitats: freshwater, brackish water and salt water. Next, second class is Phaeophyceae. Common name is brown algae. Pigments are chlorophyll A and C, fucoxanthin. The stored food is mannitol and laminaria. Cell wall is made up of cellulose and algin. Flagellar numbers are 2 and in unequal positions and lateral. Next, habitats. Freshwater is very rarely found, and saltwater marine is common in this Phaeophyceae. Next, Rhodophyceae, common name red algae. Majority pigments A and D. Phycoerythrin are the major pigments, and food stored is Floridian starch. Cell wall is made up of cellulose, pectin, and polysulfate esters. There is no flagella absent here, and habitats are freshwater, brackish water. Sexually by non-motile gametes and by sexual reproduction is oogamous and accomplished by complex post-fertilization developments. The common members are Polysyphonia, Porphyra, Gracilaria and Gelidium. Bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Next, bryophytes are called amphibians of the plant kingdom because these plants can live in soil but dependent on water for sexual reproduction. They usually occurs in damp, humid and shadowed localities. They played an important role in plant succession on bare rock soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They possess root-like, leaf-like, stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence it is called gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is antheridium. They produce biflagellated antherozoids. The female sex organ is archegonium is flask shaped and produces a single egg.
The antherozoids are released into water where it come in contact with archegonium. The antherozoids fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nutrition from it. Some cells of the sporophyte undergo reduction division, it is known as meiosis, to produce haploid spores. The spores germinate to produce gametophyte. The haploid spores to germinate to produce gametophyte. Bryophyte is generally a very little economical importance, but some mosses provide food for herbaceous mammals, birds, and other animals. The species of sphagnum and a moss provide a pet that long been used as a fuel and as a packing material for transshipment of the living material. Because of their capacity to hold water, they used as a packing material for transshipment. The mosses along with the lichens are the first organized to colonize the rock, hence are very great ecological importance. The decompose rocks making the substrate suitable for the growth of higher plants. Since the mosses form dense mats on the soil, they reduce the impact of falling rain and prevent the soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habits such as banks of streams, marshy ground, damp soil, bark of trees, and deep in woods. The plant body of liverwort is thalloid. Example, marchantia. The thallus is dorsiventral, closely appressed to the substrate, and the leafy members have thinly leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by formation of specialized structures called gymi or jama. The gymi are green, multicellular, asexual buds which develops in small receptacles called gyma cups. Located on thalli, the gymi become detached from the plant parent body and germinate to form new individual. During sexual reproduction, male and female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into foot, septa, and capsule. Foot, septa, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. Next, mosses. The predominant stage of life cycle of moss is gametophyte, which consists of two stages. The first stage is protonema stage, which directly develops from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is leafy stage, which develops from secondary protonema as a lateral bud. First one is spores, here lateral bud. They consist of upright center axis bearing spiral arranged leaves. They are attached to soil through multicellular and branched rhizoids. This stage bears sex organs. The vegetative reproduction in mosses is by fragmentation, in budding in the secondary protonema stage. In sexual reproduction, the sex organs are antheridia, archegonia, are produced at the apex of leafy shoots. After fertilization, the zygotes develops into sporophyte consisting of foot, septa, capsule. The sporophyte in mosses are more elaborated than in liverworts. The capsule containing spores are the formed after meiosis. The mosses have elaborated mechanism of spores dispersion. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Let us see some of the diagrams here. This is Marchantia female thalli identified because of the archegoniophore. Here, this is male thallus identified by antherod. Next, here, this is the downside one is gametophyte and upper one is sporophyte. Example is Funaria. This is one is Funaria. The sporophyte is divided into capsules seta. Here, gametophyte bearing the main axis, leaves and rhizoids also. Here, this one is sphagnum. It having the antheroidal branches and are pteridophytes. Pteridophytes include horsetails and ferns. Pteridophytes used for medical purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions also. You may recall that in bryophytes, the dominant phase in the life cycle is gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true roots, stems and leaves. These organs possess well differentiated vascular tissues. The leaves in pteridophytes are small, microphyles are in Selaginella. The large macrophyls are in ferns. The sporophytes bear sporangia that substitute by a leafy-like appendage called sporophyll. In some cases, sporophylls may form distinct compact structures called strobili or cones. Example, Selaginella and Equiseptum. Sporangia produces spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need of water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. Gametophytes bear male and female sex organs called antheridia and archegonia, respectively. The water is required for transfer of antherozoids, male gamete released from the antheridia to the mouth of archegonium. Fusion of male with the egg present in the archegonium results in the formation of the zygote. Thereafter produces a multicellular well-differentiated sporophyte, which is dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are similar in kind. They are known as homosporous. Genera like Selaginella salvina, which produces two kinds of spores, which are large macro and small microspores, are known as heterotrophs because two different kind of spores, hetero. The megaspores, large, and microspores, smaller, germinate to give rise to female macro and male gametophytes of micro, respectively. The female gametophytes in these plants are retained on the parented sporophytes for variable period. Development of zygotes into the young embryo takes place within the female gametophytes. This event is a precursor to the seed habit, considering an important step in evolution. The pteridophytes are further classified into four classes: Coleopsida group, Coleoptera; Lycopsida, Selaginella and Lycopodium; Spinopsida, Equiseptum; Pteropsida, including Dryopteris, Adiantum. This represents the Selaginella. Actually, it has microphyles, very small leaves, and the beside one is Equiseptum, and this is ferns, which has macrophyllus. Last one is Salvinia. Gymnosperms. The gymnosperms, gymnos means naked, sperma means seeds, naked seeds, are plants in which the ovules are not enclosed by any ovary wall and remains exposed both before and after fertilization. The seeds that develop the post-fertilization are not covered, they are naked. Gymnosperms including medium-sized trees or tall trees and shrubs. One of the gymnosperm, the giant tree, red tree, sequoia, is one of the tallest tree species. The roots generally tap roots. Roots in some genera have fungal association in form mycorrhizae, pinus, while in some other cycas, small specialized roots are called colloid roots, are associated with N2 fixing cyanobacteria. The stems are unbranched in cycas, branched in pinus and cedrus. The leaves may be simple or compound.
In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extreme temperatures, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. The thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores male and megaspore females. The two kinds of spores produced within a sporangia that borne on sporophylls which are arranged spirally along an axis to form lacs or a compact strobili or cones. The strobilis bearing microsporophylls and microsporangia are called microsporangiate or male strobili. Thus microspores develop into a male gametophytic generation which is highly reduced, is conified only a limited number of cells. This reduced gametophyte is called pollen grains. The development of pollen grains takes place within microsporangiae. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on same trees in Pinus. However, in Cycas, the male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelopes and composite structure called ovules. The ovules borne on megasporophyll which may be clustered to form female cones. The megaspore mother cell divides mitotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Like bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grains is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tubes carrying the male gametes grows towards the archegonia in the ovules and discharge their contents in near the mouth of the archegonia. Following fertilization, zygote develops into embryo and ovules into seeds. The seeds are not covered. Next, let's see the pictures. First one is named as Cycas. This is Pinus and this is Ginkgo. Dwarf shoot, long shoot and seeds. Angiosperms. Unlike the gymnosperms where the ovules are naked, in the gymnosperms are flowering plants. The pollen grains and ovules are developed in specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. The range in size from smallest Ulfia to tallest tree Eucalyptus over 100 meters. They provide us food, fodder, fuel, medicines and several other commercially important products. They are divided into two classes, dicotyledonous and monocotyledonous. The dicotyledons are characterized by seeds having 2 cotyledons, reticulate venation in leaves, tetramerous and pentamerous flowers, having 4 or 5 members in each floral whorls. The monocotyledons, on other hand, are characterized by single cotyledonous seeds, parallel venation in leaves and trimerous flowers having 3 members in each floral whorls. Next, the male sex organs in flowers is the stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grains. Microspore mature into pollen grains. The female sex organs in the flowers is pistil. Pistil consists of a swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergo meiosis to produce 4 haploid megaspores. 3 of them degenerated and 1 divide to form embryo sac. Each embryo sac 3 celled egg apertures, 1 egg cell, 2 synergids and 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. The pollen grain after dispersal from anthers are carried by a wind and various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and resulting pollen tubes grow through the tissues of the stigma and style, reaching the ovule. The pollen tube enters the embryo sac when 2 male gametes are discharged. One of the male gametes fuses with the egg cell known as syngamy to form zygote. The other male gamete fuses with the diploid secondary nucleus to produce triploid primary endosperm called PEN. PEN. Because of the occurrence of the 2 fusions, syngamy and triple fusion, this event is termed as double fertilization. And in event unique to 2 angiosperms, the zygote develops into an embryo with 1 or 2 cotyledons and the pen develops into endosperm which nourishment to the developing embryo. The synergids and antipodals degenerated after fertilization. During the events of the ovules develop into the seeds and ovaries develops into the fruits, and the life cycle of the angiosperm is shown in the figure. Let's see the figure here. The microspores, nothing but pollen grains, which is reaching the megasporophyll ovary through pollinations and development of the pollen grains in the stigma and all reaching the ovary. The both double fertilization is going to happen and the formation of zygote to embryo to sporophyte. Here you can see that the sporophyte is a diploid generation 2n, gametophytic is haploid. The fusion and the fertilization restores the number and it becomes the diploid. Here you can see the female gametophyte with style stigma on the top and the base of the swollen one known as ovary, and it also has megaspore mother cells, megasporangium ovules. Here upper one is filament which releases a microsporangium pollen grains. Again it is all similar one. Pollination occurs, fertilization, and again formation of zygote to a formation of endosperm and embryo. It is a repeated process, less like a cycle. Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. The haploid spores are produced by this plant body by meiosis. There is in turn divided by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. 1. Sporophytic generation is represented only by one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form gametophyte. The dominant and photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvax, spirogyra and some species of chlamydomonas represents this pattern, haplontic or haploid you can say. On the other extreme is a type where diploid sporophyte is dominant and photosynthetic independent phase of the plant. The gametophytic phase is represented by a single-celled haploid gametophyte. This life cycle is termed as diplontic. In alga, fucus represents this pattern. In addition, all seed-bearing plants, gymnosperms and angiosperms follow this pattern with some variations. The bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however difference in dominant phase. A dominant and independent photosynthetic thalloid or erect phase is represented by haploid gametophyte, and it's alternate with short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. Diploid sporophyte is represented by a dominant and independent photosynthetic vascular plant body. It alternates with multicellular sporophyte and autotrophic independent but short-lived haploid gametophyte. Such pattern is haplodiplontic cycles. All pteridophyte represents this pattern. Interestingly, with algae genera are haplontic. Some of them are Ectocarpus polyciphonia, and kelps are haplodiplontic. Fucus of alga is diploid. Let us see some of the patterns of the figures here. Haplontic. Here the gametophyte, gametogenesis, syngamy, and the zygote is diploid 2n. Again through meiosis it produces the spores. The spores will be the haploid. Coming to diplontic, here the sporophyte is 2n. Through meiosis it produces spores, which is haploid. Gametophyte and gametogenesis, again syngamy restores it. This phase haplodiplontic, which is represented by bryophytes and pteridophytes. In bryophytes the dominant phase is gametophyte, and in pteridophytes the sporophyte is the dominated one.